Welcome back to the deep dive. Today we are opening a file that honestly reads a bit like a script for a high-stakes pandemic thriller.
It really does.
Yeah, I mean, we aren't talking about the common cold or a simple tummy bug today. We are stepping into what I like to call the sci-fi villain wing of microbiology.
It sounds a little dramatic when you phrase it that way, but you really aren't wrong. If you were a writer trying to design the ultimate biological threat, you couldn't come up with better mechanisms than what we see in the families we are covering today.
Right, the Bunyaviridae and the Arenaviridae.
Exactly, those two.
Because we're talking about viruses that literally steal hats from your own cells to disguise their genetic material.
The cap-snatching mechanism, yeah.
Right, and we're talking about viruses that carry actual grains of sand inside their bodies, which turn out to be stolen parts of the host. And of course, pathogens that turn the human immune system against itself in just the most violent way possible.
We are dealing with absolute masters of disguise here, and critically, masters of zoonosis.
Zoonosis, the jump from animal to human.
Right. These aren't human viruses that just, hang out in the coffee shops or daycares. These are animal viruses that live in completely parallel worlds.
The insect world, the rodent world.
Yes. And they only cause devastation when we accidentally cross into their territory. Today is really all about that crossover event.
So for the medical students, the residents, and, just the lifelong learners tuning in, here is our mission for you today. We are breaking down Chapter 53 of Murray's Medical Microbiology. That's the 9th edition.
That's our structural spine for this deep dive.
Exactly. Right? But we aren't just stopping there. We've also pulled in the ecological data from Chapter 38 of Jaywitt's.
Which is really necessary to fill in the gaps on how these things actually get to you in the real world.
Because the goal here isn't just to memorize a list of bugs. That's the old way of studying. And frankly, it just doesn't stick.
No, it doesn't. You forget it the second the exam is over.
Yeah.
We want you to build a conceptual framework. You need to understand segmented genomes, why they actually matter for viral evolution.
Right.
And you need to understand the specific ecological niches these viruses exploit. If you understand the ecology, the clinical presentation just automatically starts to make sense.
It totally does. So here is the roadmap for what we're doing. We're going to start with the massive Bunyaviridae supergroup. We'll look at their rule of three structure, this absolutely crazy cap-snatching heist they pull off, and the big divide between the bug-borne arboviruses and the rodent-borne hantaviruses.
And then after that, we'll pivot over to the Arenaviridae.
The sandy one.
The sandy ones. We'll explain that morphology, the absolute brain twister that is essence genetics, and the heavy hitters like Lassa fever.
And as always, we will wrap up with a consolidation session, a rapid-fire quiz to make sure you can actually answer the board questions when they pop up.
Sounds like a solid plan.
One quick disclaimer before we get into the weeds, though. Taxonomy in virology changes, it changes faster than fashion trends.
It's a complete mess of reclassification right now. If you look at the latest phylogenetic papers, names are shifting, families are splitting.
There are hundreds of viruses involved here.
Right. But for the purpose of your exams, and specifically following Murray's 9th edition, we are sticking to the classic groupings.
Yeah, we want your mental shelves to be organized, not cluttered with this week's debate on viral nomenclature.
Precisely. Let's keep the mental model clean. You can argue about phylogeny after you pass the boards.
Deal. So let's unpack the Bunyaviridae. And the first thing that hits you when you open the book is the sheer size of this family. Murray literally calls it a supergroup.
Because it is massive. We are talking about over 200 distinct serotypes.
Wow.
It is arguably the largest group of RNA viruses that we deal with in medical microbiology.
That number has to be terrifying for a student. Memorize these 200 viruses?
No, don't let the number scare you. You don't need to know all 200. You just need a system to organize them.
Okay.
And structurally, this whole family shares a very specific architectural rule. I call it the rule of three.
I love a good rule. Break down the rule of three for Bunyaviridae.
So it refers to their genome. These are negative sense RNA viruses. But unlike, say, rabies or measles, which just have one long continuous strand of RNA bunyaviruses, have a genome that is segmented into 3 distinct pieces.
3 pieces, like a 3-volume encyclopedia.
Exactly. And they are named very creatively. The L segment, which stands for large.
Okay.
The M segment for medium, and the S segment for small.
Large, medium, small. Microbiologists are very literal people.
We try to be. But this segmentation is crucial for understanding them. Think about influenza. We always talk about antigenic shifts with the flu because it has 8 segments.
Right, so if two different flu viruses hit the same cell,
They can swap segments and create a completely new Frankenstein virus.
genetic reassortment.
Exactly. Bunyaviruses can do the exact same thing because of this rule of 3. If 2 strains infect the same mosquito or the same human, they can shuffle those L, M, and S cards.
Which allows for rapid evolution and new strains popping up.
Yes. So that's the first pillar of our framework. 3 segments. Now, there is another structural detail that Murray highlights as a major exam differentiator. It has to do with the skeleton of the virus, or rather, the lack of one.
Okay, let's hear it.
Most negative sense RNA viruses have something called a matrix protein. It's a structural shell that sits just inside the viral envelope. It acts like a frame. It makes the virus rigid and uniform.
Like the poles in a tent?
Perfect analogy. The matrix protein is the tent poles. But bunyaviruses generally lack a matrix protein.
No matrix protein. So using the tent analogy, they are a tent without poles.
Essentially, it makes them pleomorphic.
Pleomorphic meaning they take on multiple shapes.
Right. They don't have a rigid, uniform shape. They are a bit floppy, a bit variable in size. But, and this is critical, they are enveloped.
I want to pause on the word enveloped. We hear that word a lot in virology, enveloped versus naked capsid. But in terms of transmission, it's a huge clue for you as a student, isn't it?
It is the single most important clue for prevention and epidemiology. An envelope is just a lipid membrane. It's fat.
Right.
What happens to fat when it dries out? It degrades.
Yeah.
What happens when you hit it with soap or detergent?
It dissolves.
Exactly. So enveloped viruses are wimps in the environment.
They are fragile. They cannot survive on a doorknob or a table surface or a dry hand for very long.
They need moisture to maintain that lipid bilayer. They need a wet ride.
A wet ride. I really like that phrase.
They need to go directly from fluid to fluid, blood to blood, or mucous membrane to mucous membrane. They cannot survive the dry wasteland of the outside world like an adenovirus or a norovirus can.
Which totally explains the transmission routes. That's why the vast majority of these are arboviruses, transmitted by arthropods like mosquitoes and ticks. They are literally injecting the fluid directly into your blood.
Or in the case of hantavirus, through moist rodent excreta. But you aren't going to catch these from a casual handshake or touching a dry surface.
Got it. So 3 segments. Enveloped, which means fragile. No matrix protein, which means floppy. Now let's get to the heist. You mentioned these viruses steal hats. This is the molecular mechanism that always blows my mind.
This is cap snatching. It's
Yeah.
one of my absolute favorite mechanisms in biology because it is so ruthlessly efficient.
Set the scene for us. Why do they need a hat in the first place?
Okay, so the bunyavirus is a negative sense RNA virus. That means its genome is the opposite of messenger RNA, or mRNA. Can't be read by the ribosome immediately.
So it carries its own polymerase, right? The L protein to make mRNA.
Yes, it brings its own photocopier.
Mhm.
But here's the problem. The host cell, your cell, is very protective of its ribosomes. It has a security system. The ribosome will only bind to mRNA if it has a specific ticket at the front end.
And that ticket is a methylated 5' cap.
Exactly. The host cell's own mRNA has this VIP pass. No pass, no entry to the ribosome club.
So if the virus just makes raw RNA without a cap, the ribosome just ignores it.
Totally ignores it. So what does the bunyavirus do? It doesn't have the machinery to make its own cap. That requires complex cellular enzymes it doesn't carry.
Right.
So its polymerase literally grabs a piece of the host's own mRNA, and it cleaves off the first 10 to 18 bases, which includes that 5' cap.
It decapitates the host's message.
It decapitates the host RNA and uses that stolen cap as the primer to start writing its own viral RNA.
That is just astonishing. It's literally like stealing someone's ID badge at the front door of an office building.
It is identity theft at a molecular level, and it serves 2 major purposes. 1, it allows the viral RNA to be translated by the host, and 2, because it is destroying the host's caps, it shuts down the host cell's ability to make its own proteins.
So it cripples the cell while boosting itself.
Exactly. Talk about kicking someone when they're down.
Now, here is the high-yield nuance for the test takers out there. Influenza also does cap snatching. How do we tell them apart on a board question?
Location, location, location. Influenza replicates in the nucleus, so it snatches caps in the nucleus. Bunyaviruses replicate in the cytoplasm.
Okay, so bunyaviruses are snatching these caps in the cytoplasm. It's a cytoplasmic heist.
Correct. That is a great differentiator. Influenza is nucleus. Bunyavirus is cytoplasm. Both are thieves, but they rob different banks.
Now, let's talk about how they actually leave the cell once they replicate, because most enveloped viruses, once they're built, they just push their way out through the cell membrane, right? The outer skin.
Yes, usually. But bunyaviruses are hipsters. They have to do it differently.
They bud into the Golgi apparatus.
They do. They bud into the Golgi.
That's deep inside the cell. That's the shipping and receiving center.
Right, the viral glycoproteins, the spikes that will eventually be on the outside of the virus. They don't travel to the outer cell membrane immediately like other viruses. They accumulate in the membranes of the Golgi.
So the virus basically assembles inside the factory itself.
It assembles in the Golgi, buds into the Golgi vesicles, and then those vesicles travel to the surface and release the virus.
Why does it matter practically? Is it just a trivia point for an exam?
It matters for detection. If you look at an infected cell under a microscope, you wouldn't necessarily see viral spikes all over the outer surface of the cell immediately, because they're hiding inside the Golgi until the last possible second.
Sneaky, it helps them hide from the immune system a bit longer.
Precisely. So if you see a question about an RNA virus budding into the Golgi, you should immediately think Bunyaviridae.
Okay, so we have the structure down. We have the rule of three, the fragile envelope, the cap snatching in the cytoplasm, and the Golgi budding. Now let's talk about how these viruses actually make people sick. This brings us to the two roads of pathogenesis.
Yes, this is the best way to divide the family mentally. Because there are so many of them, we can divide them based on transmission. Road 1 is the arboviruses. Road 2 is the roboviruses.
Let's walk down road 1 first, arboviruses.
This includes the genera Bunyavirus, Flabovirus, and Nairovirus. Arbo stands for arthropod-borne, mosquitoes, ticks, sandflies, the biters. The cycle here is classic. The bug bites you. Inject the virus into your blood. You get a primary viremia, meaning virus circulating in the blood.
And that usually feels like the flu, right?
Yes. Fever, aches, general malaise. For many people, the immune system actually wins right here, and it stops.
But for the unlucky ones,
The virus disseminates. It finds a target organ. Depending on the specific virus, that might be the central nervous system, which causes encephalitis, or the liver, causing necrosis, or the vascular endothelium, causing hemorrhage.
I want to touch on the ecology here because Giles brings up a really creepy detail about the vectors. These viruses have a problem. Mosquitoes die in the winter.
Right. Imagine
Yeah.
winter in Ohio or Wisconsin. A hard freeze. All the adult mosquitoes die. So where does the virus go? Does it just disappear?
You would certainly hope so.
Unfortunately, no.
Mhm.
Some of these viruses, like the lacrosse virus, have evolved transovarial transmission.
Transovarial, meaning through the ovaries.
Yes. The female mosquito passes the virus directly into her eggs. Those eggs are laid in tree holes or old tires. And they can actually survive the freezing winter.
They just sit there dormant and infected.
Exactly. When spring comes and the rain fills those tires with water, the eggs hatch. The larvae that come out are already infected with the virus.
That is incredibly persistent. The virus just hibernates in the babies.
It ensures the virus survives from season to season without ever needing a mammalian host. The mosquito isn't just a bus for the virus. It's a reservoir. It maintains the virus in nature.
That is road 1, arboviruses. Now let's look at road 2, the roboviruses. This is the hantavirus.
Robo for rodent-borne. Hantavirus is the definitive black sheep of the Bunyaviridae family
Why is it the black sheep?
because it is the only bunyavirus that does not use an arthropod vector. There's no mosquito, no tick, no sandfly. It relies entirely on rodents.
So if it doesn't have a mosquito to inject it into your blood, how does it get from the mouse to the man?
Aerosolization. This is the dusty shed scenario. The rodent, which is usually a deer mouse for the sin number virus, sheds the virus in its urine, its feces, and its saliva.
And mice pee basically everywhere.
They do. That waste dries up on the floor. It mixes with the dust. Then you walk in to sweep the shed, or you're hiking and you kick up some dirt in a cabin. You create an aerosol cloud of virus-laden dust.
And you just inhale it deeply into your lungs.
Right into the lungs. And the damage mechanism is very different, too. With the arboviruses, we talked about cell death, cytolysis. But hantavirus is different.
Pathogenesis for hantavirus is an immunopathologic storm.
That's right. It sounds violent because it is. The virus infects the endothelial cells, the lining of your blood vessels, specifically in the lungs. But it doesn't actually burst them open. It doesn't kill the cell directly.
So the virus is relatively quiet.
The virus is, but your immune system is not. Your immune system detects the virus and reacts with massive, overwhelming force.
It releases huge amounts of cytokines.
Yes. These cytokines tell the blood vessels to become permeable, to open up the gaps between the cells, to let immune cells in.
But in the lungs, Leaky blood vessels are an absolute disaster.
Exactly. Fluid rushes out of the blood and into the alveolar space. You literally drown in your own plasma. That is hantavirus pulmonary syndrome.
So it's essentially friendly fire. The virus isn't destroying your lungs. Your own immune response to the virus is flooding them.
Correct. And that helps explain why the disease moves so fast, which we will discuss in the clinical section next.
Let's dig into those clinical syndromes right now. We have the mechanisms down. Let's look at the patients. Start with the arbovirus side, the encephalitis. The big one for American medical students is Lacrosse virus.
Lacrosse is the most common cause of pediatric arboviral encephalitis in the U.S. as part of the California encephalitis group.
Give us the classic clinical vignette. Who is the patient?
You are looking for a child, usually school age, say 10 to 14 years old. It is July or August. They live in the Midwest, Ohio, Wisconsin, Minnesota, or maybe the Appalachian region.
And what's the risk factor?
They've been playing outside in the woods.
Yeah.
But specifically, look for the mention of discarded tires.
Why tires? Is it just because kids like playing on tires?
It's because the mosquito loves them.
Yeah.
The vector is Aedes triceriatus, the eastern tree-hole mosquito.
Right.
In nature, it breeds in water-filled holes in trees. But old tires are basically artificial tree holes. They hold stagnant rainwater. They are dark. They are warm. It's a mosquito luxury condo.
So we have a 12-year-old kid in Ohio in the summer playing near tires. What are the symptoms?
It starts out very nonspecific. Fever, headache, maybe some vomiting. But then there's a fast progression to CNS involvement. Lethargy, confusion, and the big red flag seizures.
Seizures are a hallmark.
Very prominent. About 50% of hospitalized cases have seizures. It presents as a severe meningoencephalitis.
Is it highly fatal?
Rarely. The mortality is actually less than 1%.
Yeah.
But, and this is very important for counseling parents, the neurological sequelae can linger.
So they don't just bounce back perfectly.
No. Some kids have persistent seizure disorders or learning difficulties for years after recovery. It is certainly not a benign infection.
Okay, let's contrast that with the hantavirus clinical picture. This is hantavirus pulmonary syndrome, or HPS. The story here usually starts with the SIN number outbreak.
The virology history books love this one. The year was 1993. The location was the Four Corners region of the U.S., where Utah, Colorado, Arizona, and New Mexico all meet.
Usually a very dry desert region.
Usually, yes. But that year, there had been unusually heavy rainfall due to an El Niño weather pattern. The rain made the pinyon trees bloom like crazy. There was a massive crop of pine nuts.
And pine nuts mean very happy mice.
Exactly. The deer mouse population, Paramyscus maniculatus, just exploded. It was a plague of mice. More mice naturally means more contact with humans.
And then healthy young people started dropping dead.
That was the terrifying part for public health officials. These weren't frail or elderly patients. These were young, athletic people.
What were their symptoms?
They would come in with what felt like a bad flu. Myalgia, fever, maybe some GI symptoms. But then, within 24 to 48 hours, they would just crash.
When you say crash.
I mean catastrophic respiratory failure.
Yeah.
Their lungs filled with fluid. They went into severe shock. The mortality rate in that initial outbreak was over 50%. Even today, with modern ICU care, it's still around 30 to 40%.
That speed is terrifying, from I feel a bit achy to full intubation in a single day.
That's the cytokine storm at work. Once that tap opens, the lungs flood incredibly fast. And here's a key diagnostic clue for the boards.
Let's hear it.
Because the virus doesn't kill the cells directly, the lung biopsy will show viral antigen in the endothelium, but there will be no necrosis.
No necrosis. That is a great differentiator. Now, you also mentioned a geography split here, New World versus Old World hantavirus.
Yes. What we just described, HPS, the lung issue, is the New World presentation in the Americas. But in Europe and Asia, the Old World, hantavirus looks very different.
This is the Hantan virus, which was originally found in Korea.
Right. In the Old World, the primary target organ is the kidney. It causes HFRS, hemorrhagic fever, with renal syndrome.
So New World equals lungs, Old World equals kidneys plus bleeding.
Correct. The Old World patient presents with fever, then shock, and then acute kidney failure. They stop making urine. It's still a vascular leak issue, but the leak is happening in the renal bed rather than the lungs.
That's a crucial distinction for exams. If the patient is in Korea, you look at the creatinine levels. If they're in New Mexico, you look at the oxygen saturation.
Exactly right.
All right, that covers the Bunyaviridae. We have the rule of three, the Golgi budding, the tire-breeding mosquitoes, and the mouse dust lung failure. Now, let's shift gears to the second family, the Arenaviridae.
The Arenaviridae, and the name is actually the first clue here. It comes from the Latin word arena, which means sand.
Which brings us back to the hook from the beginning. Why are they called sandy?
When early virologists looked at these viruses under an electron microscope, they were genuinely confused. They saw the viral envelope, but inside, they saw these grainy, dense, sand-like particles.
What were they?
They turned out to be host cell ribosomes.
Wait, the host cell's own ribosomes?
Yes. The virus, during assembly, accidentally packs the host's own ribosomes into its capsid.
That seems really sloppy.
It is sloppy. As far as we know, those ribosomes are completely non-functional. They are just junk luggage. The virus doesn't use them at all.
But they are the defining visual feature.
Exactly. If an exam question mentions a sandy appearance on electron microscopy or host ribosomes inside the virion, you can stop reading. It is an arenavirus.
Okay, sandy equals arenavirus. Now, bunyaviruses had the rule of 3 segments. What about arenaviruses?
They follow the rule of 2. They have 2 RNA segments, L for large and S for small.
Just L and S. That's simple enough.
The number is simple. The coding strategy is not. This is one of the most complex concepts in RNA virology. The arenavirus S segment is ambiscent.
Like ambidextrous. It goes both ways.
Precisely. This is where students always get tripped up, so let's really break it down. Usually, a negative sense RNA virus is just a template. It has to be copied into positive sense mRNA to make a protein, one direction.
Right. Negative makes positive. Positive makes protein.
But the arenavirus S strand is weird. The first half of the strand is negative sense, but the second half is positive sense.
How does the virus even handle that? It sounds like a car trying to drive forward and in reverse at the exact same time.
It handles it by creating a timing mechanism, a 2-phase production line.
Walk me through the phases.
Phase 1. The virus enters the cell. The polymerase reads the negative part of the strand immediately, and it makes mRNA for the nucleocapsid protein.
So step 1 is basically build the internal machinery.
Right. But it cannot read the other half yet. That positive sense part is tucked away in a secondary structure, usually a tight hairpin loop. The ribosome can't even see it.
Okay, so how do we get to phase 2?
The virus has to replicate the entire genome first. It makes a full-length genome copy. Now, that second half, which was positive, becomes negative in the copy. Now the polymerase can read it.
That is a serious brain bender.
Think of it as a security lock. The virus forces itself to replicate its genome before it can make the glycoproteins for the outer shell.
Ah, so it separates the guts, the nucleocapsid, from the skin, the glycoprotein, by time.
Exactly. It ensures that you have plenty of internal guts ready to go before you start manufacturing the outer skin to package it all up. It's a brilliant temporal regulation strategy.
That is incredibly sophisticated for a sloppy packer that carries around junk ribosomes.
Evolution is full of contradictions.
It really is. Okay, so rule of 2, sandy ribosomes, ambisense coding. What about the reservoir? We talked about rodents for hantavirus.
Arenaviruses are also strictly rodent-borne, but the relationship is even tighter. These viruses establish lifelong persistent infections in their specific rodent hosts.
So the mouse is infected for its entire life.
For life. And importantly, the mouse doesn't get sick. It creates a state of immune tolerance. It just sheds virus in its urine, feces, and saliva forever.
That sounds like a perfect evolutionary setup. The host stays alive to spread the virus?
It is. And because different rodent species live in different parts of the world, the geography of the virus is completely determined by the geography of the rat.
Which leads us nicely to the clinical syndromes. Let's start with the one we might see here in the US or Europe, LCMV.
Lymphocytic choriomeningitis virus. The reservoir here is the common house, mouse, musculus, and also hamsters.
Hamsters. This is the pet store angle.
Yes. There have been several documented outbreaks linked to pet store chains or hamster breeding facilities. If you have a vignette about a new pet hamster and a flu-like illness, think LCMV.
What does the illness actually look like?
It's biphasic, meaning 2 phases. Phase 1 is the viral replication phase. Fever, malaise, muscle aches. The virus is circulating in the blood.
And then what?
Then the patient often gets better for a few days. The fever breaks. They think they're fine.
The false dawn.
Exactly. Then comes phase 2. The fever returns, along with a severe headache, stiff neck, and photophobia. This is an aseptic meningitis.
What is happening in phase 2 is the virus attacking the brain.
This is the fascinating part. Phase 2 is immunopathologic. It's not the virus eating your brain. It's your T cells attacking the virus-infected cells in the meninges.
How do we actually know that?
We've done experiments with mice. If you take a mouse that lacks T cells, so it's immunocompromised, and infect it with LCMV, it won't get the meningitis.
Really?
Yes, it will have a ton of virus replicating in its brain, but no inflammation, no symptoms, It dies eventually from overall organ failure, but it doesn't get the acute meningitis.
That is wild. The immune response is the disease.
Correct.
There is also a very specific warning for pregnant women regarding LCMV, right?
Major warning. It acts essentially as a tear or seed pathogen. It is highly teratogenic. It can cause hydrocephalus and choroidinitis blindness in the fetus. Pregnant women should strictly avoid cleaning mouse droppings or handling hamsters for this reason.
Okay, so LCMV is the meningitis one. Now let's talk about the truly scary one. Lassa fever.
Lassa is a viral hemorrhagic fever. It is a massive public health issue in West Africa. Nigeria, Sierra Leone, Liberia, Guinea.
What's the reservoir for Lassa?
The mastomys rat, also known as the multi-mammoth.
Multi-mammoth. Sounds like a hyperproductive rodent.
It is a very prolific breeder, which really helps the virus spread. It lives in and around human homes in rural West Africa.
Lassa is often compared to Ebola in the news. Is it as deadly?
Not quite. The mortality of hospitalized cases is around 15 to 20%, whereas Ebola can be upwards of 50 to 90%. But Lassa is far more common. We see hundreds of thousands of infections a year.
How does it present clinically?
Gradual onset. Fever, vomiting, retrosternal pain, which is chest pain. But then it progresses to facial swelling, bleeding from the gums, internal bleeding, and profound shock.
There is a very specific long-term effect of Lassa fever that shows up on exams constantly.
Deafness, sensorineural hearing loss.
Why deafness specifically?
We think the virus damages the auditory nerve directly. About 25% of patients, even those with mild disease who didn't get the severe hemorrhagic shock, lose their hearing. Sometimes it comes back partially, but often it is permanent. It's a hallmark sequela.
Deafness. Okay, lock that in. And just to round out the geography, we have the South American cousins.
Right. Junin virus in Argentina, Machupo virus in Bolivia. These are also arenaviruses, also rodent-borne, and they cause very similar hemorrhagic fevers.
And the risk factor here is often agricultural, isn't it?
Yes. These viruses live in field mice, the kind that infest corn and wheat fields. So harvest season always brings outbreaks. In Argentina, Junin virus is known as Argentine hemorrhagic fever, and it historically affected the maize harvesters.
Geography is destiny with these viruses. If you are in a cornfield in Argentina, it's Junin. If you are in a village in Nigeria, it's Lassa. If you are cleaning a shed in New Mexico, it's Sin Nombre.
Exactly. The virus is wherever the specific rodent is.
Let's move to Section 6. Diagnosis, treatment, and prevention. How do we spot these things without getting killed in the process?
Diagnosis for both families relies heavily on RT-PCR. We want to detect the viral RNA. Serology IgM and IgG is useful later, but in the acute phase, PCR is the gold standard.
And what about safety? We talked about how Lassa is scary.
Critical. Arenaviruses, especially Lassa and Machupo, are biosafety level 4 pathogens, BSL-4.
Define BSL-4 for us.
That is the highest possible level of containment. We are talking about positive pressure moon suits with completely independent air supplies, multiple airlocks, dedicated negative pressure rooms. You do not try to culture Lassa fever in a standard hospital lab. If you do and you create an aerosol, you could infect the entire lab staff.
So if you suspect Lassa, you call the CDC or the Specialized Infectious Disease Unit immediately. Don't be a cowboy with the Petri dish.
Never be a cowboy with hemorrhagic fevers.
What about treatment?
For most, it is just supportive care. Fluids, oxygen, managing blood pressure. But there's one antiviral drug that comes into play here, ribavirin.
Ribavirin. We hear about this for hepatitis C sometimes.
Right. Ribavirin is a guanosine analog. It messes with the viral RNA polymerase. It causes lethal mutagenesis. It makes the virus make so many typos in its genome that the virus essentially collapses.
When do we use it?
It's highly effective for Lassa fever if it's given early. It dramatically reduces mortality. It also has some efficacy for the old world hantaviruses, HFRS, and potentially Junin and Machupo.
But not for the New World hantavirus.
Sadly, no. Ribavirin has not shown benefit for hantavirus pulmonary syndrome Sin Nombre, so for the lung version in the U.S., it's strictly supportive care. ECMO, or extracorporeal membrane oxygenation, might be needed to breathe for the patient while the lungs recover.
And prevention, since we don't have widely available vaccines for most of these.
It comes down to breaking the chain of transmission. For hantavirus and arenavirus, that means aggressive rodent control. Seal the food, trap the mice, and this is highly practical advice, never sweep a dusty shed with a broom.
Because it aerosolizes the virus.
Exactly. Wet mop it with bleach. Don't stir up the dust. For the arboviruses like lacrosse, it's vector control. Bug spray, screens on windows, and getting rid of those old tires where the mosquitoes breed.
Okay, we have built the framework. We have covered the structure, the bugs, the rats, and the clinical signs. Now it is time to test your recall.
Let's see if the concept's stuck.
This is our rapid-fire consolidation round. I'll give you a clinical vignette or a fact. You shout out the answer or whisper it if you're commuting, and then our expert will break it down. Ready?
Ready.
Question 1. A 12-year-old boy in Ohio is brought to the ER in July with sudden onset fever, vomiting, and seizures. His parents mention he spends all day playing in a wooded area near a pile of old tires. Which virus is the most likely culprit? Take a second.
The answer is lacrosse virus. It's a member of the Bunyaviridae. Why? Because the geography of Ohio and the Midwest, the season summer, and the specific risk factor of discarded tires breeding Aedes triceriatus point directly to lacrosse encephalitis. The seizures are the key clinical feature that distinguishes it from a simple flu.
Perfect. Question 2. A man cleaning out a dusty shed in New Mexico develops sudden, severe shortness of breath. A lung biopsy shows viral antigens in the endothelium, but notably, the cells are not dead. There is no necrosis. Instead, there is massive fluid leakage. What is the viral reservoir?
The answer is the deer mouse, Paramescus maniculatus. This is hantavirus pulmonary syndrome caused by the sin nombre virus. The location in the southwest and the activity of cleaning a dusty shed are classic. The mechanism is an immunopathologic vascular leak, not cytolysis, which is why you see the no necrosis finding on biopsy.
Spot on. Question 3. You are looking at an electron micrograph of a virus. It has an envelope and 2 RNA segments, but the most striking feature is a sandy appearance caused by grainy particles inside the capsid. What is the unique coding strategy of its S segment?
The answer is ambison's coding. The sandy appearance instantly identifies it as an arenavirus. Arena means sand. Arenaviruses follow the rule of 2L and S segments, and the S segment uses an ambison's coding strategy where part is positive and part is negative.
Awesome. Question 4. A medical volunteer returns from Sierra Leone complaining of fever and a sore throat. She quickly deteriorates into hemorrhagic shock. She is treated with ribavirin and survives, but during recovery, she realizes she cannot hear out of her left ear. What is the pathogen?
The answer is Lassa fever virus. The geography of West Africa, the hemorrhagic presentation, and the very specific sequela of sensorineural deafness identify Lassa. The fact that ribavirin was effective is also a major clue.
Excellent. Question 5. We talked a lot about viral assembly today. Which specific cellular organelle do bunyaviruses use for their envelope assembly and budding?
The answer is the Golgi apparatus. Most enveloped viruses bud straight from the plasma membrane. Bunyaviruses are unique in that they bud into the Golgi cisternae before being transported to the cell surface.
All right, last one. Question 6. Unlike the rest of the Bunyaviridae family, hantaviruses lack which specific mode of transmission?
The answer is arthropod vector transmission. Hantaviruses are roboviruses, rodent-borne, not arboviruses. They do not use mosquitoes or ticks. They rely entirely on aerosolized rodent excreta.
How'd you do? If you got those, you are in great shape for the boards.
And you have successfully navigated the sci-fi wing of the viral zoo.
Before we sign off, let's just zoom out. We have these 2 families, Bunyaviridae and Arenaviridae. What is the big takeaway here?
The unifying theme is absolutely zoonosis. These viruses live in parallel worlds, the insect world, the rodent world, and they only cause devastation when we accidentally cross into their territory, whether it is old tires, a dusty shed, or a cornfield in Argentina. Human behavior and geography dictate the risk.
And here is a final provocative thought to leave you with. We talked about how arenaviruses pack host ribosomes into their capsid, those grains of sand. It's basically junk luggage. Evolutionarily, it just seems like a mistake.
It does. They don't seem to use them for anything at all.
But if the virus has a mechanism to package large, complex cellular machinery like a ribosome inside itself, could we hack that? Could we design a drug or a genetic payload that looks like a ribosome to the virus so it accidentally packages its own poison pill?
A Trojan horse strategy using the virus's own assembly error. That is a truly fascinating idea. If we could figure out the precise signal that triggers that packaging, we might be able to weaponize their sandy nature against them.
Something for you to mull over. For now, your call to action is simple. Read Murray chapter 53, look at the maps. Geography is destiny for these viruses, so visual memory really helps.
And check those Javits tables for the vector specifics if you want that extra edge on the exam.
Thanks for listening to The Deep Dive. Wash your hands, avoid the mouse droppings, and we'll catch you on the next one.
Stay curious.